Hello students, this is Kanika. Welcome to my channel Prep Biology. So today we will continue with the unit 1, that is reproduction. We have started and this is the class 2nd of this series. So today in this lecture we will start with very important topic of this chapter, that is the structure of stamen and the section of the stamen, that is the TS of a anther. Let's start with the lecture. We know that there are two parts of the stamen. Stamen is the male reproductive part. Stamen contains filament and anther. As you can all see here that this is the anther part and this is the filament part. This we have covered in the previous lecture. So, today we will elaborately study about the structure of the stamen and section of the anther. Now, we know that the anther and the filament is the part of the stamen. Now, anther is bilobed structure. This term is very important. Bilobed means, bi means two, having two lobes. What does this mean? So, anther contains, this is the one lobe and this is the second lobe. Okay. So, this is the one lobe and this is the second lobe. That's why the anther is bilobed structure. Okay. And second term, which is very important, in angiosperms, the anther is dithecous in nature. Dithecous in nature. Dithecous means having two thecas. Having two theca. Okay. So, understand it carefully. This is one lobe and this is the second lobe. Okay. This is one lobe and this is the second lobe. One and second. One lobe contains two theca and second lobe also contains two theca. Theca means it has two compartments or chambers inside it. Chambers or compartments inside it. Is it clear? So, how many total theca are present in one anther? 2 plus 2, that is 4 theca in one anther. Is it clear? We have two lobes of anther. If we cut the section transversely, transverse means across the length, that is the horizontally, we have this kind of structure. So, this is one lobe and this is second lobe. This is one lobe and this is the second lobe. Okay. Inside each lobe, we have how many compartments? We have two compartments in each lobe. Okay. Those compartments are called the theca. Those compartments are called the theca. This is one lobe. If you see here, this is one lobe and inside one lobe we have, this is 1 and 2 theca and this is second lobe and this is 1 and second theca. So, that's why anther in angiosperms, the anther is dithecous or bithecous in nature. Is it clear to you all? This is very important. And if we talk about the filament is a cylindrical stalk-like structure, slender stalk-like structure. This is the stalk of the anther and the proximal part of the filament, that is the basal part of the filament, it is attached to the petal of a flower or thalamus of a flower. This point is also important. Is it clear? Now let's understand from this picture. This is the structure of anther, and we know this is the filament and this is the anther. We cut the section horizontally or transversely, that is the TS of anther. We have how many total chambers inside it? We have 4 chambers inside it. Okay, and these chambers contains a number of pollen grains. When it gets mature, it contains number of pollen grains. Okay, so let's start with very important topic, that is the TS of anther. TS of anther. If we cut these are the lobes of anther. If we cut the anther like this, that is transversely, that is called the TS, transverse section of anther. So this section is very important for your exam point of view. So understand it carefully. If we cut this, we have this kind of structure. This is the one lobe. We have this kind of structure. Okay, this is first lobe and this is the second lobe. Now inside the lobes, we know how many compartments in each lobe? Two compartments in each lobe, that is called dithecous nature. If we see this largely, so it has this kind of structure. The anther contains tetragonal structure. Anther contains tetragonal structure like this. It has 4 sides. Tetragonal, tetra means 4 sides, and inside it we have 4 sacs are present. So inside the anther we have microsporangia. Microsporangia or sporangium is there. How many microsporangia are there in one anther? There are total 4. 1, 2, 3 and 4 microsporangia are there in one anther. Okay, so is it clear to you all? Now let's see the TS of anther. Firstly, we will discuss about the transverse section of young anther. When the anther is young, then the structure is seen like this under the microscope. So anther contains, we know, 4 sacs. Each sac contains 4 microsporangium. Okay, microsporangium we used for single microsporangium. If microsporangias are more than one, then we use microsporangia. Is it clear? So microsporangium is singular term and microsporangia are plural term. Okay, so let's see. The anther is covered by 4 walls. This is very, very important. The anther is covered by 4 walls. These walls are very important. So outermost wall, look at here, the outermost wall of an anther is called the epidermis. Inner to epidermis, which is shown by the green pen, that is called the endothecium. This is the second layer. First layer is epidermis, second layer is endothecium. Inner to endothecium, we have 3 to 4, this is 1, 2, 3, 3 to 4 layers. Those layers are called the middle layer. Is it clear? This is the third layer. And the last layer, innermost layer, that is the tapetum. This layer is very, very important because it nourishes the pollen grains. Okay, so first is the epidermis, second is endothecium, third is middle layer, and fourth one is the tapetum. And tapetum surrounds the microsporangium. Is it clear? Tapetum surrounds the microsporangium. If we talk about the young anther when it is not matured, then microsporangium contains special kinds of cells. These are called the sporogenous cells. Okay, in young anther, the sporogenous cells are present. What are sporogenous cells? These are the compactly arranged homogeneous cells. These are the same kind of cells. Homo means same kinds of cells are present inside the microsporangium. I hope this is clear. Now, if we see the microscopic view, this picture shows the microscopic view. Inside the microscope, when we cut the section of the anther, that is the transverse section of the anther, under the microscope, this kind of structure is seen. Look at here, there are the multiple layers. We have discussed how many layers are surrounds the anther, that is the 4 layers. Outermost is epidermis, inner to epidermis is endothecium, inner to endothecium is middle layer, and the innermost layer is the tapetum. Okay, and this is the structure of the mature anther. Is it clear? Because the pollen grains are formed, that's why it is called the mature anther. And if you can see here, this kind of structure is seen at the middle of the anther. This is called the connective. Connective is a part where anther lobes, this is important, note it down, anther lobes fused together. Okay, so if you have a question that in which part the anther lobes are fused together, that part is called the connective part. Okay, so this is important. Let's study about the different layers of anther and its function. Okay, so you can take screenshot of this chart and add it in your notes. This is the most simplified way of representing the different layers. First one is outermost is epidermis, second one is endothecium, third one is the middle layer, and fourth is tapetum. Okay, let's see the number of each layer. Epidermis is single layered. Okay, this is a single layer, one layer is present. Endothecium is also single layered, but middle layer is about 3 to 4 layers. Okay, this is important. And tapetum is also single layered. Now, if we see the composition of each layer, this composition of each layer is very important. Epidermis. Epidermis is outermost layer and it is thickened. It is made up of thickened fibrous matter. Okay. They have fiber-like thickenings are present inside it. Second one is the endothecium. Endothecium is radially elongated cells that develops cellulose with pectin and lignin. So understand it carefully. The endothecium, if you can see here, this is the epidermis, single layered. Endothecium is this kind of structures, cells are present inside the endothecium and they are radially elongated, okay, and they develop cellulose. Cellulose, as you all know, this is the polymer, and with the pectin and the lignin. Pectin and lignin are the also polymers. They are the polymers, okay, and they are the main constituent of the cell walls of many plants. Okay, they are the main constituents of cell walls of many plants. Pectin is polysaccharide, that is the carbohydrate made up of multiple monomers of carbohydrate, and the lignin is also a organic polymer. This is very important. So which layer forms, develop cellulose with pectin and lignin? That is the endothecium. Okay. Now next layer is middle layer. Middle layer is thin walled. This is very important. This is thin walled and epidermis is thick walled. Okay. Middle layer is thin walled and the tapetum, this layer is very important. It is close to the pollen sac because it is innermost. It is multinucleate. Multinucleate means it has multiple nucleus, dense cytoplasm. It has dense cytoplasm, and why it has dense cytoplasm? Because it is rich in mitochondria and ribosomal vesicles. These 4 points are very important. Tapetum is multinucleate, dense cytoplasm, rich mitochondrial and ribosomal vesicle. Now let's see the function of each layer. First is the epidermis. Epidermis, it helps in the production and structure integrity. It maintains the structure of an anther. Okay. Second one is the endothecium. Endothecium, it prevents from splitting and bursting of pollen sac. That is the microsporangium. Okay. It helps in preventing the bursting of a pollen sac. Next is the middle layer. Middle layer helps in the pollen production, and the tapetum is for nourishment. It nourishes the developing pollen grains and provide the nourishment. So this topic is very important for your board, 12th board exam point of view. In every year you have 1 to 2 questions, around 1 to 2 questions from this topic. So TS of anther and their anther walls, okay, and their function, their composition is very important. So make sure this topic is clear to you all. Okay, so this is all about today's lecture. I hope this lecture is beneficial for you all. If you like this lecture, please like, share and subscribe my channel, that is Prep Biology. Thank you so much.